Hello, and welcome to another journey through the gentle wonders of our natural world. Tonight, we're going to explore the quiet, mysterious realm of mushrooms. These fascinating organisms live just beneath our notice, hidden in the forest floor, tucked into the bark of trees, spreading through the soil in networks we can barely imagine. As you settle into your pillows and let your breathing slow, we'll wander together through 100 facts about these remarkable beings. Let your mind drift with the words, and if sleep finds you before we reach the end, that's perfectly fine. The mushrooms will still be there tomorrow, growing silently in their secret places. Take a deep breath. Feel yourself sinking deeper into your bed. Tonight's journey will take us into forests and fields, into microscopic worlds and continental networks, through time and across evolution. We'll discover organisms that glow in the dark, giants that span forests, healers and destroyers, partners and solitary wanderers. Let the words wash over you like a gentle rain on forest leaves. Mushrooms are neither plants nor animals. This is our first fact, and it's fundamental to understanding everything that follows. They belong to their own kingdom, entirely the kingdom fungi, which stands separate from the plant, animal, and bacterial worlds. For most of human history, people assumed mushrooms were plants. After all, they grow from the ground, they don't move, they seem rooted in place. But in 1869, scientist R.H. Whitaker proposed that fungi deserved their own kingdom in the classification of life, and subsequent research proved him right. Scientists have identified about 148,000 species of fungi so far, ranging from single celled yeasts to massive underground networks. But they estimate that somewhere between 1.5 million and over 5 million species may actually exist on Earth. Think about that for a moment. For every fungal species we know, there might be 10 or even 30 species still waiting to be discovered. Most of these remain completely unknown to us, living their lives in soil and wood, in the deepest parts of oceans, in polar ice, unseen and unstudied, each one with its own particular way of making a living in the world. The diversity is staggering. There are fungi that eat insects, fungi that partner with algae to create lichens, fungi that digest oil spills, fungi that grow inside other fungi that can withstand intense radiation, fungi that survive in the driest deserts and the coldest glaciers. The kingdom fungi encompasses this extraordinary variety while sharing certain fundamental characteristics that unite them all. Before we go much further, I'm curious, where in the world are you listening to this right now? Are you tucked into bed in a city apartment? Or perhaps in a house surrounded by trees? Maybe you're in a place where mushrooms grow wild in the autumn rains, or perhaps in a climate where they're rare and precious. I always love imagining where you all are, scattered across this planet we share with these extraordinary organisms. Wherever you are, I hope you're comfortable and ready to let these gentle facts about mushrooms carry you toward rest. The word fungus comes from the Latin word for mushroom, and indeed, the familiar mushroom is what most people picture when they think of fungi. But mushrooms are actually just the fruiting bodies of much larger organisms. Think of them as the apples on an apple tree. The real body of the fungus, called the mycelium, lives underground or inside wood, spreading out in vast networks of thread-like structures called hyphae. These hyphae are so fine, so delicate, that thousands of them could fit on the head of a pin. Each hypha is a tubular cell, sometimes containing many nuclei within a single continuous cytoplasm, sometimes divided by walls called septa. These hyphae grow at their tips, extending ever outward, branching and rebranching, exploring the environment in search of nutrients. The growing tip of a hypha secretes enzymes that break down complex organic molecules in the surrounding substrate, whether that's soil, wood, leaf litter, or even animal dung. Once the molecules are broken down into simpler sugars, amino acids, and other compounds, the hypha can absorb them through its cell wall. This external digestion is one of the defining characteristics of fungi. They digest their food before eating it, in a sense. As hyphae spread and branch, they form a network called the mycelium. Picture a three-dimensional web, spreading through soil or within a rotting log. This web can be incredibly dense in some areas and sparse in others, depending on where nutrients are concentrated. The mycelium is the main body of the fungus, the part that lives and grows for years or decades or even millennia. It's mostly invisible to us because it's hidden in the substrate it's feeding on. Mushrooms are more closely related to animals than they are to plants. This discovery surprised scientists when genetic analysis revealed it in recent decades. For centuries, fungi were classified as plants. They seemed to have so much in common with the green world, sessile, growing from the ground, or on wood, appearing to have roots. But DNA analysis told a different story. The genetic code of fungi shows that they share a more recent common ancestor with animals than with plants. This means that you are more closely related to a mushroom than a mushroom is to a rose. Like us, fungi cannot photosynthesize. They cannot capture the sun's energy and turn it into food. They don't have chlorophyll, those green molecules that allow plants to harvest light. Instead, they must consume other organisms or organic matter to survive, making them heterotrophs, just like animals. They break down organic material and absorb the nutrients, feeding on the world around them. The cells of fungi even contain chitin in their cell walls, the same substance that forms the hard exoskeletons of insects and the shells of crustaceans. Touch an insect's shell. That hardness, that slightly plasticky feel, that's chitin. The same substance forms the walls that surround fungal cells, giving them structure and protection. Plant cell walls, by contrast, are made of cellulose. This shared chemistry between fungi and arthropods hints at an ancient connection, a kinship that goes back hundreds of millions of years to when our ancestors and the ancestors of fungi diverged along different evolutionary paths. Fungal cells also use ergosterol in their cell membranes, a steroid molecule that functions like the cholesterol found in animal cell membranes. This similarity is why some antifungal medications work by targeting ergosterol, disrupting the fungal cell membrane without harming animal cells. The molecular machinery inside fungal cells, how they produce energy, how they synthesize proteins, how they regulate their genes, all of this bears the marks of their relationship to the animal kingdom. Most fungi spend their lives as mycelium, that underground network of threads. The mycelium can live for years, decades, even centuries, spreading slowly through soil and wood. It feeds continuously, absorbing nutrients, growing ever outward. During this time, it might never produce a single mushroom. It simply exists, quiet and patient, doing the work of breaking down dead matter, sometimes forming partnerships with plant roots, cycling nutrients through the ecosystem. The mushrooms we see above ground appear only when conditions are right for reproduction. They emerge when temperature and moisture align, when the mycelium has accumulated enough resources, when the season is appropriate for spore dispersal. It's as if the mycelium has been saving up, gathering strength, and then suddenly decides the time is right. Tiny knots of hyphae form in the mycelium, condensing and organizing into structures called primordia. These primordia are baby mushrooms, pin-sized at first, barely visible. When conditions remain favorable, these primordia develop rapidly. Water from the mycelium flows into the developing mushroom, inflating its cells like balloons being filled. The cap unfolds from the stem. The gills or pores develop underneath. The protective veil tears away. Within hours or days, a fully formed mushroom stands where there was nothing visible before. This mushroom might live for just a few days, releasing its millions or billions of spores into the wind. Then it shrivels, decays, returns to the earth. But the mycelium remains, patient and persistent, waiting for the next opportunity to fruit. This pattern, long-lived mycelium producing short-lived fruiting bodies, is the fundamental rhythm of mushroom life. The mycelium is the marathon runner, pacing itself for the long haul. The mushroom is the sprinter, appearing suddenly, performing its reproductive function intensely and quickly, then disappearing just as suddenly. The largest living organism on Earth is not a whale or a giant sequoia. It's not even anything you can see all at once. It's a fungus. In the Malheur National Forest in eastern Oregon, in the Blue Mountains, beneath the pines and firs, spreading through the forest floor like an invisible net, lives a single specimen of Armillaria ostoyae, commonly called honey mushroom. This one individual organism spreads across 3.5 square miles of forest floor. That's 2,385 acres, the size of 1,665 football fields laid end to end. Imagine walking through this forest on an autumn day. The air is cool, smelling of pine needles and damp earth. Here and there, you might notice clusters of honey-colored mushrooms sprouting from the base of a tree or emerging from the soil. These mushrooms don't look particularly impressive. They're modest in size, with caps a few inches across, pale yellowish brown with darker scales. You might walk past them without a second glance. But beneath your feet, spreading through the soil in every direction, connecting all these scattered mushroom clusters, is a single living organism, one individual fungus that has been growing for thousands of years. Scientists determined through genetic testing that all the mushrooms fruiting across this enormous area belong to one individual organism. They collected samples from mushrooms and mycelium from many different locations across the forest and analyzed their DNA. What they found was that samples taken from locations miles apart were genetically identical. They were all part of the same individual, cloned from the same original spore that germinated thousands of years ago. This massive fungus is estimated to weigh between 7,500 and 35,000 tons. depending on how much of the underground biomass you include in the calculation. To put that in perspective, 7,500 tons would be the weight of about 50 adult gray whales. 35,000 tons would be equivalent to more than 200 gray whales. Either way, we're talking about an organism of almost incomprehensible mass. The organism is thought to be between 2,000 and 8,650 years old. Think about what that means. If it's 8,000 years old, it started growing during the Neolithic period, when humans were first developing agriculture in the Fertile Crescent. It was already ancient when the pyramids of Egypt were built. It has lived through the entire span of recorded human history, and long before. It has survived fires, droughts, ice ages, the arrival and departure of countless animal species. It has simply kept growing, slowly, steadily, spreading through the forest soil at a rate of 1 to 3 feet per year. The honey fungus spreads through the forest at a rate of about 1 to 3 feet per year, sending out dark, root-like structures called rhizomorphs that search through the soil for new sources of food. These rhizomorphs can travel considerable distances before finding a suitable tree root to colonize. The fungus feeds on both living and dead wood, and while it's considered a destructive forest pathogen because it kills trees, it also plays a vital role in breaking down dead organic matter and recycling nutrients back into the ecosystem. Another enormous fungus was discovered in Michigan in 1992. This armillaria specimen covers about 37 acres and weighs approximately 440 tons, equivalent to 3 blue whales. It originated from spores that traveled from Ontario, Canada, and has been growing for about 2,500 years. These discoveries fundamentally change how scientists think about the size and lifespan of organisms. Fungi are incredibly diverse in their forms and lifestyles. Some are single-celled, like yeasts, which have been used for thousands of years to leaven bread and ferment beer and wine. Others form complex, multicellular structures. Mushrooms can range from tiny caps, barely larger than a pinhead, to giant puffballs the size of soccer balls. The variety in shapes, colors, and textures seems almost endless. Mushrooms reproduce by releasing spores instead of seeds. These spores are microscopically small, typically between 3 and 12 microns in diameter. A micron is 1,000th of a millimeter. To give you a sense of scale, a human hair is about 70 microns in diameter. So these spores are many times smaller than the width of a human hair. They're so small that they're essentially invisible individually. When you see mushroom spores, you're seeing them in vast multitudes as a powder or a print. A single mushroom can produce billions of spores. A common field mushroom, for instance, can release 16 billion spores over the course of a few days. 16 billion. That's more than twice the number of people on Earth, all produced by one mushroom about the size of your fist. A giant puffball can release 7 trillion spores. 7 trillion. The mind struggles to comprehend such numbers. But these astronomical figures make sense when you consider that the vast majority of spores will never find suitable conditions to germinate and grow. Most will land in places where they cannot survive, where the temperature is wrong, where moisture is absent, where competing organisms are too strong. Spores drift through the air like invisible smoke. They're so tiny and light that even the gentlest breeze can carry them. Turbulent air keeps them aloft. They can rise high into the atmosphere, traveling on wind currents for enormous distances. Spores from wheat rust fungi, for instance, have been documented traveling over 1,200 miles through the air, crossing from continent to continent. A spore that lands in your garden might have begun its journey in a forest hundreds of miles away, carried by storms and prevailing winds, floating through clouds, settling finally to earth when the air stills. Water also plays a role in spore dispersal. When raindrops strike a mushroom cap, they can splash spores into the air like tiny wet explosions, each droplet carrying spores outward from the parent mushroom or washing them along the ground. Some mushrooms time their spore release to coincide with rain, maximizing the chances that water will help disperse their offspring. Animals contribute too, often unknowingly. A deer walking through the forest brushes against a mushroom, and thousands of spores adhere to its fur. Miles away, the deer lies down to rest, and the spores fall off, finding new territory. Birds, landing on mushrooms, carry spores on their feet and feathers. Insects, crawling across gill surfaces, pick up spores on their legs and bodies. Even slugs and snails, slowly munching on mushroom flesh, inadvertently become spore vectors, depositing spores in their trails of slime or in their droppings. The mechanism by which mushrooms release spores is itself remarkable, a tiny feat of biological engineering that happens millions of times across each mushroom. In Basidiomycete, mushrooms, which include most of the familiar gilled mushrooms you see in forests and lawns, spores are ejected from the gill surface with incredible precision. Each spore sits on a tiny stalk called a sterigma, projecting from a specialized cell called a basidium. As the spore matures, a tiny droplet of water, smaller than the spore itself, condenses on its surface. This is called Buller's drop, named after the mycologist who first described it. At the same time, another tiny droplet forms on the surface of the sterigma, just below the spore. When these two droplets make contact and merge, something remarkable happens. The sudden coalescence of the droplets changes the distribution of weight and creates a brief but intense momentum. This catapults the spore away from the gill surface at an acceleration of over 10,000 times the force of gravity. Imagine experiencing 10,000 Gs of acceleration. Fighter pilots train to withstand 9 Gs for brief moments. Astronauts during launch experience 3 or 4 Gs. This spore, this microscopic particle, smaller than a mote of dust, launches itself with an acceleration that would turn a human into paste. It travels fast enough and far enough to clear the narrow gap between adjacent gills, falling into the air current below the mushroom cap. The spacing between gills is critical. If they're too close together, spores ejected from one gill will hit the neighboring gill and stick there, wasting the effort. If they're too far apart, the mushroom uses more energy than necessary to build its structure. Evolution has fine-tuned gill spacing to match the exact distance that spores can be ejected. It's a beautiful example of optimization in nature, millions of years of natural selection refining the architecture to maximize efficiency. But mushrooms don't rely entirely on external winds for dispersal. Research has shown that mushrooms create their own airflows to carry spores away. As moisture evaporates from the mushroom's surface, it cools the air immediately surrounding the cap. This cooler air is denser and sinks, creating a gentle convective current that flows downward and then outward from beneath the cap. These self-generated currents can carry spores several centimeters per second and lift them 10 centimeters or more into the air, where they can catch larger air movements. This discovery helps explain how mushrooms can successfully disperse spores even in environments with very little wind, such as the understory of dense forests. Spores that land in suitable locations can germinate, sending out thin threads of hyphae that begin to explore their environment. These hyphae secrete enzymes that break down complex organic molecules in the soil, wood, or other substrate, allowing the fungus to absorb the resulting simpler nutrients. The hyphae grow and branch, gradually forming a network, the mycelium, that can cover large areas. For sexual reproduction to occur, hyphae from two different mating types must meet and fuse. Fungi can have many different mating types, far more than the simple male and female of animals. Some species have thousands of different mating types, which greatly increases the chances that any two individuals that meet will be compatible. When compatible hyphae fuse, they form a new mycelium that contains genetic material from both parents. This mycelium eventually produces mushrooms, which generate and release spores, continuing the cycle. Fungi have existed for an extraordinarily long time. Fossil evidence suggests that fungi were among the first organisms to colonize land, appearing about 800 million years ago, long before plants made the transition from water to land. Some scientists believe that fungi may have actually helped plants colonize land by forming partnerships with early plant roots, just as they do today. These partnerships, called mycorrhizal associations, are one of the most important ecological relationships on Earth. Between 80 and 90% of all plant species form mycorrhizal associations with fungi. This statistic is worth pausing over. Nearly every plant you see, every tree in the forest, every shrub in your garden, every wildflower in the meadow is partnered with a fungus underground. The plant's roots are wrapped in or penetrated by fungal hyphae, creating an intimate connection at the cellular level. In these relationships, the fungal hyphae wrap around or penetrate plant roots, creating what's essentially a trading interface between two different organisms. The fungus uses its extensive network of hyphae, which can explore a much larger volume of soil than plant roots can, to gather water and mineral nutrients from the surrounding earth. Hyphae are much thinner than the finest root hairs, allowing them to penetrate tiny pores in the soil that roots cannot reach. They produce enzymes that can liberate nutrients locked up in mineral compounds or organic matter. The fungus gathers up phosphorus, nitrogen, and other essential nutrients and transports them to the plant roots, delivering them in forms the plant can readily absorb. Some fungi can even access nutrients from rocks, slowly dissolving minerals over time. In exchange, the plant provides the fungus with sugars produced through photosynthesis. These sugars, created from sunlight, water, and carbon dioxide, represent captured solar energy in chemical form. The fungus, which cannot photosynthesize, depends entirely on the plant for this energy source. This is not a casual partnership. For many plants, mycorrhizal fungi are absolutely essential for survival. Without these fungal partners, the plants cannot grow properly or survive at all. They simply cannot gather enough nutrients from the soil on their own. Orchids provide a dramatic example. Orchid seeds are extraordinarily tiny, among the smallest seeds in the plant kingdom. They're little more than an embryo wrapped in a thin seed coat, containing almost no stored energy, no reserves to draw upon. When an orchid seed germinates, it cannot photosynthesize yet because it has no leaves. It must immediately form a relationship with a fungus that will provide it with nutrients, essentially feeding the baby orchid until it can grow leaves and begin making its own food through photosynthesis. Some orchids remain dependent on fungal partners throughout their entire lives, even as adults, receiving nutrients from the fungus rather than making all their food through photosynthesis. The mycorrhizal network connecting plants through fungal hyphae has been called the Wood Wide Web, a term that's caught the public imagination because it so perfectly captures what researchers have discovered about how these networks function. This name was popularized by German forester Peter Wohleben in his book about trees, though the scientific study of these networks began much earlier. Through this underground network, trees and other plants appear to communicate and share resources in ways that challenge our traditional understanding of how forests work. Research by forest ecologist Suzanne Simard at the University of British Columbia has revolutionized our understanding of forest ecosystems. Her work has shown that large, mature trees, which she calls mother trees, can transfer carbon to smaller, younger trees through shared mycorrhizal networks. These mother trees, the old giants of the forest, are not just passively connected to the network, they actively support younger trees, especially their own offspring, sending them extra nutrients through the fungal connections. Simard's research has shown that these mother trees can actually recognize their own offspring, their own seedlings, and direct more resources toward them than toward unrelated seedlings. How they recognize kin remains a subject of active research, but it appears to involve chemical signals in the soil and through the mycorrhizal network itself. This discovery fundamentally changes how we think about forests. Trees aren't just individuals competing for light and nutrients. They're part of communities, families even, supporting each other through underground networks. The networks can extend across enormous areas, connecting hundreds or even thousands of plants of many different species. A single mycorrhizal network in a forest might include Douglas firs, paper birches, cedars, ferns, and dozens of other plant species, all linked through the fungal threads in the soil. The fungus doesn't discriminate. It connects with any compatible plant root it encounters, creating a web that spans the forest. When one plant is attacked by insects or infected by disease, it can send chemical warning signals through the network. These chemical messages travel through the fungal hyphae to neighboring plants, allowing them to activate their defenses before the threat reaches them. A tree being eaten by caterpillars might send signals that cause nearby trees to start producing defensive chemicals, making their leaves less palatable, even though they haven't been attacked yet. This gives them a head start in defending themselves. Plants under stress, perhaps suffering from drought or nutrient deficiency can receive support from healthier neighbors through nutrient transfers via the network. A tree that's shaded and struggling to photosynthesize might receive carbon from a tree in a sunny spot. These transfers can mean the difference between survival and death for struggling plants. The fungi themselves benefit enormously from maintaining this network. By keeping their plant partners healthy, the fungi ensure a steady supply of the carbon rich sugars they need to survive and grow. In exchange for these sugars, the fungi retain about 30% of what the plants produce through photosynthesis. It's a significant cost for the plants, but the benefits in terms of nutrient acquisition and stress resistance make it worthwhile. This arrangement works so well that mycorrhizal networks have been operating for hundreds of millions of years, since plants first colonized land and needed help establishing themselves in soil. But not all fungi are helpful partners. Some are parasites that attack living plants, animals, or even other fungi. The honey fungus we mentioned earlier, that enormous organism in Oregon, is actually a forest pathogen. It attacks and kills trees by growing under the bark, at the base of the trunk, and around the roots, eventually girdling the tree and cutting off the flow of water and nutrients. While this might seem entirely destructive, the honey fungus also helps break down dead wood and recycle nutrients, playing a role in forest renewal and succession. Other parasitic fungi have even more specialized lifestyles. Cordyceps fungi infect insects and other arthropods. When a cordyceps spore lands on an insect, it germinates and begins growing into the insect's body. The fungus eventually kills the host and produces a fruiting body that erupts from the insect's corpse, often from the head. These fungal fruiting bodies can be quite dramatic, sometimes looking like small, antler like structures emerging from a dead ant or caterpillar. Some cordyceps species can even manipulate their host's behavior, causing infected ants to climb to elevated positions before dying, which helps the fungus disperse its spores more effectively. Fungi's role as decomposers might be their most important function in ecosystems. Without fungi, dead plant and animal matter would accumulate endlessly. Forests would choke on fallen leaves and branches. Nutrients locked up in dead tissue would never return to the soil. Fungi are uniquely equipped to break down even the most resistant plant materials. They produce powerful enzymes that can digest cellulose, the main component of plant cell walls, and lignin, the tough, woody substance that gives trees their strength and rigidity. Very few other organisms can decompose lignin effectively. As fungi break down dead material, they release nutrients back into the soil where plants can absorb them again. This decomposition is part of the essential nutrient cycles that keep ecosystems functioning. Carbon, nitrogen, phosphorus, and other elements move from living organisms to dead matter to soil to living organisms again, cycling endlessly. And fungi are key players in maintaining these cycles. Some fungi have evolved to decompose surprisingly specific materials. There are fungi that specialize in breaking down bird feathers, others that focus on animal droppings, and still others that consume fallen pine needles or decaying oak leaves. This specialization means that different types of organic matter decompose at different rates, depending on which fungi are present and active in an ecosystem. Humans have used fungi for thousands of years without really understanding what they were. The 5,300-year-old mummy, known as Ötzi the Iceman, found frozen in the Austrian Alps in 1991, was carrying two species of polypore mushrooms. One, Fomes fomentarius, also called the tinder fungus, was likely used as tinder for starting fires. This mushroom, when dried and processed, catches sparks easily and smolders slowly, perfect for carrying fire from place to place or starting new fires. The other mushroom Ötzi carried, Piptoporus betulinus, called the birch polypore, has antimicrobial properties and may have been used medicinally. Some researchers believe he was using it to treat intestinal parasites, as evidence of parasitic worms was found in his remains. Ancient peoples used fungi in food preparation, often without realizing they were working with living organisms. Fermentation, which relies on yeasts, has been used for millennia to make bread, beer, and wine. The ancient Egyptians were baking leavened bread as early as 3,000 years ago, relying on wild yeasts to make their dough rise. They didn't know about microorganisms. They just knew that if they left dough sitting out, it would bubble and expand, and when baked, it would be lighter and tastier than unleavened bread. The Chinese have used mushrooms in traditional medicine for at least 2,000 years, valuing certain species for their health promoting properties. Ancient texts describe mushrooms as promoting longevity, enhancing vitality, and treating various ailments. Reishi mushrooms, known in China as lingji, were reserved for emperors and their families, considered too precious for common people. These mushrooms, growing on decaying trees in mountain forests, were sought after as medicines and symbols of good fortune. Today, we know that mushrooms contain many bioactive compounds with potential health benefits. Medicinal mushrooms, like reishi, turkey tail, lion's mane, chaga, and cordyceps, have been studied extensively in modern laboratories for their effects on the immune system, inflammation, cognitive function, and even cancer. While more research is always needed, and while mushroom supplements should never replace conventional medical treatment, preliminary studies suggest that compounds found in these mushrooms, particularly polysaccharides called beta-glucans, may help modulate immune responses and provide antioxidant benefits. Reishi mushrooms, Ganoderma lucidum, known as the mushroom of immortality in traditional Chinese medicine, have been used for over 2,000 years. Ancient Chinese texts rank them among the most cherished superior herbs, those thought to support long life, boost energy, and promote overall health. These mushrooms contain over 400 different bioactive compounds, including beta-glucans and triterpenoids. Research suggests that these compounds may help lower blood sugar and blood pressure levels, potentially reducing the risk of diabetes and heart disease. Some studies indicate that reishi mushrooms may increase white blood cell activity, potentially strengthening immune function and helping the body fight infections. Turkey tail mushrooms, Tremites versicolor, get their name from their appearance: colorful, concentric rings on fan-shaped fruiting bodies that resemble a wild turkey's tail feathers. They grow on dead logs and stumps worldwide. These mushrooms contain a compound called polysaccharide K, or PSK, which has been approved as a cancer treatment adjunct in Japan since the 1970s and in China since the 1980s. Clinical studies suggest it may help strengthen the immune system in cancer patients undergoing chemotherapy or radiation therapy, potentially improving outcomes. In China, at least 12 turkey tail based drugs are currently approved for clinical use. While these mushrooms aren't a cure for cancer, they represent an area of ongoing research into how natural compounds might support conventional cancer treatments. Lion's mane mushrooms, Hericium erinaceus, have an unmistakable appearance. Unlike typical mushrooms with caps and stems, lion's mane looks like a white waterfall of icicles or a shaggy mane, hence the name. They grow on hardwood trees, particularly oak and maple. These mushrooms have attracted significant attention for their potential effects on brain health and cognitive function. They contain compounds called hericenones and erinacines that may stimulate the production of nerve growth factor, or NGF, a protein important for the growth, maintenance, and survival of neurons. Some preliminary research suggests that lion's mane may support memory and cognitive function and might protect against neurodegenerative conditions, though much more human research is needed to confirm these effects. Chaga, Inonotus obliquus, grows primarily on birch trees in cold climates like Alaska, northern Canada, Siberia, and northern Europe. It doesn't look like a typical mushroom. Instead, it forms black, charred-looking masses on the outside of birch trunks, looking more like burnt wood than a living organism. Inside, the tissue is rusty brown. Chaga is extraordinarily rich in antioxidants, containing more antioxidants per gram than most other foods. A single serving of chaga extract contains as many antioxidants as 30 pounds of carrots. These antioxidants, including compounds like melanin and superoxide dismutase, may help protect cells from damage caused by free radicals and oxidative stress, processes implicated in aging and various diseases. Traditional medicine in Russia and Northern Europe has used chaga for centuries to treat various ailments and promote general health. But while some mushrooms offer health benefits, others are deadly poisonous. The death cap mushroom, Amanita phalloides, is responsible for 90% of all mushroom-related fatalities worldwide. This one species, this one type of mushroom, kills more people than all other poisonous mushrooms combined. As little as half a mushroom, about 50 grams of fresh tissue, contains enough toxin to kill an adult human. For a child, even less would be lethal. The death cap contains a group of toxins called amatoxins, particularly alpha-amanitin. These are among the most potent natural poisons known. Alpha-amanitin works by inhibiting RNA polymerase II, an enzyme absolutely essential for protein synthesis in cells. RNA polymerase II reads DNA and creates messenger RNA, which cells then use as instructions for making proteins. Without this enzyme functioning, cells cannot produce the proteins they need to survive. Without protein production, cell metabolism grinds to a halt, and the cell dies. The toxin is particularly damaging to the liver and kidneys, organs with high metabolic rates that are exposed to high concentrations of the poison as it passes through the body. The liver, which processes toxins from the bloodstream, receives the full force of the amatoxin. Liver cells begin dying within hours of ingestion. The kidneys, filtering blood to produce urine, also concentrate the toxin and suffer severe damage. What makes the death cap particularly dangerous? What gives it an especially sinister quality is that symptoms don't appear immediately. After eating death cap mushrooms, a person might feel fine for 6 to 24 hours. During this latent period, they might congratulate themselves on a successful forage, might enjoy their meal without any apparent ill effects, might go to bed thinking all is well. But during this deceptive calm, the toxins are circulating through the bloodstream, entering cells, binding to RNA polymerase, shutting down protein synthesis, killing cells by the billions. Then, suddenly, severe gastrointestinal symptoms begin: cramping, nausea, vomiting, and diarrhea strike with violence. The person becomes severely dehydrated. This phase can last for 24 to 48 hours. It's absolutely miserable. But then, strangely, the symptoms may improve. The person might feel somewhat better. The vomiting stops. The diarrhea eases. They might think the worst has passed, that they're recovering. But this improvement is an illusion, a cruel false hope. During this apparent recovery, which doctors call the deceptive improvement phase, the liver and kidneys are failing. Toxins continue destroying these organs from within. 2 to 5 days after ingestion, jaundice appears as the liver stops functioning properly. The skin and eyes turn yellow. Blood tests show skyrocketing liver enzymes. Kidney function deteriorates. Without a liver transplant, performed quickly, death often follows within a week of eating the mushroom. Even with modern medical care, even with intensive support, the mortality rate remains around 10 to 15%. The death cap is native to Europe, where it's been known for millennia. Ancient Greeks and Romans knew it was deadly. It may have been used in several famous assassinations. Roman Emperor Claudius possibly died from death cap poisoning in AD 54, though historical records are unclear. Some scholars believe his wife Agrippina served him death caps mixed with edible Caesar's mushrooms. Holy Roman Emperor Charles VI may have died from death cap poisoning in 1740. Throughout history, these mushrooms have been tools of murder, nearly undetectable and almost always fatal. In recent decades, the death cap has spread far beyond Europe. It's been introduced to other continents, likely transported on the roots of imported trees, particularly oaks. European trees planted in parks and gardens in North America, Australia, South Africa, and New Zealand have brought their mycorrhizal fungal partners with them, including Amanita phalloides. The mushroom now grows in California, Oregon, Washington, and British Columbia, where it fruits under oaks in late autumn and winter. It's established in Australia, particularly around Melbourne and Canberra. It's spreading in these new environments, forming associations with local tree species. The mushroom can vary considerably in appearance, which makes identification challenging and dangerous. The cap is typically greenish yellow or olive colored, but it can range from pale yellowish tan to brown, or even white in some varieties. White death caps are particularly treacherous because people might mistake them for edible puffballs or meadow mushrooms. The cap starts out rounded like an egg, then expands to become convex or nearly flat, typically 5 to 15 centimeters in diameter. The gills underneath the cap are white and free, not attached to the stem. The stem is white often with a slight greenish tinge, and it has a prominent ring around it, a collar of tissue that remains from the veil that protected the gills as the mushroom developed. At the base, the stem swells into a bulb that sits inside a cup-like structure called a vulva, the remains of the universal veil that enclosed the entire young mushroom. This resemblance to several edible mushroom species has led to many tragic poisonings. In Asia, death caps are sometimes mistaken for edible straw mushrooms, which are popular in cooking. In Europe, people have confused them with Caesar's mushrooms, which are choice edibles. In North America, white forms have been mistaken for meadow mushrooms or puffballs. Even experienced foragers have made fatal mistakes. The rule among mushroom hunters is absolute. You must be 100% certain of identification before eating any wild mushroom. If there's any doubt whatsoever, don't eat it. It's simply not worth the risk. Other deadly mushrooms include the destroying angel, another Amanita species with pure white mushrooms, and various species in the genera Galerina, Lepiota, and Conocybe, which also contain amatoxins. The autumn skullcap, or deadly webcap, contains different toxins that cause kidney failure. Even experienced mushroom foragers exercise extreme caution, following the rule that they must be 100% certain of a mushroom's identity before eating it. Interestingly, animals seem to have different tolerances for mushroom toxins. Squirrels and rabbits can eat death caps without apparent harm, which has sometimes led people to make the dangerous assumption that if animals can eat a mushroom, humans can too. This is absolutely not true. What is safe for one species may be deadly for another. Not all poisonous mushrooms are deadly. Some cause severe gastrointestinal distress, but rarely lead to death. Others cause neurological symptoms, including hallucinations. Psilocybin mushrooms, often called magic mushrooms, contain compounds that affect serotonin receptors in the brain, leading to altered perceptions and hallucinations. These mushrooms have been used in spiritual and religious ceremonies by various cultures for thousands of years. In recent years, researchers have begun studying psilocybin for its potential therapeutic effects in treating depression, anxiety, and PTSD, though these remain controlled substances in most countries. Some mushrooms glow in the dark. This might sound like something from a fairy tale, but it's absolutely real. About 125 species of bioluminescent fungi have been discovered so far, mostly in tropical and subtropical regions, though some occur in temperate forests. These mushrooms produce light through a chemical reaction involving a light-emitting compound called luciferin and an enzyme called luciferase. When luciferin is oxidized by luciferase in the presence of oxygen, it releases energy in the form of light. It's the same basic process that fireflies use, though the specific molecules involved are different. The light produced by bioluminescent mushrooms is typically a greenish glow with a wavelength around 520 to 530 nanometers. This is in the middle of the visible spectrum, the color of spring leaves or new grass. The intensity is usually quite dim, requiring very dark conditions to see. You wouldn't notice it during the day. Even at dusk, the glow might be too faint to detect. But in the deep darkness of a forest, at night, when your eyes have had time to adjust, When the moon is down and clouds cover the stars, then you might see it, a soft, ethereal glow emanating from rotting logs, from clusters of mushrooms growing on a tree trunk, from the forest floor itself. Imagine walking through a rainforest at midnight. The darkness is absolute, thick enough to feel. You've turned off your flashlight, letting your eyes slowly adapt. Gradually, you begin to see tiny points of light, like earthbound stars scattered across the forest floor. As your eyes adjust further, you realize you're surrounded by glowing fungi. Threads of light trace the outlines of fallen logs. Mushroom caps shine with gentle radiance. The mycelium itself seems to be luminous, creating patterns of light in the dark. Different parts of different mushroom species glow. In some, it's only the cap or the gills that emit light. In others, only the stem produces the glow. In many species, the mushroom doesn't glow at all, but the mycelium, those thread-like networks, underground or within wood, shines brightly. This is the phenomenon known as foxfire, a glow that appears on rotting wood in dark forests. People have reported foxfire for centuries, sometimes calling it fairy fire. They knew that certain types of decaying wood would glow at night, though they didn't understand the mechanism. Armillaria mushrooms, including the massive honey fungus we discussed earlier, that enormous organism in Oregon, can display bioluminescence in their mycelium. Imagine that 3 square-mile network glowing faintly in the darkness of the forest, an underground light show that no one sees unless they dig into the soil or find a piece of colonized wood. Why do mushrooms glow? Scientists have proposed several hypotheses, and the answer might be different for different species. For mushrooms, where the fruiting body glows, the light may attract nocturnal insects that then help disperse spores. It's like how flowers use bright colors to attract daytime pollinators, except mushrooms are using light to attract nighttime spore dispersers. Studies have shown that bioluminescent mushrooms do indeed attract more insects than non-glowing controls. Beetles, flies, wasps, and ants gather around the glowing mushrooms. As they crawl over the surfaces, they pick up spores on their bodies and carry them away. For species where only the mycelium glows, the purpose is less clear. The light might attract predators of insects that feed on the mycelium, providing the fungus with a form of protection. An insect munching on glowing mycelium becomes visible in the darkness, making it easier for predators to spot and capture. Or perhaps the glow serves as a warning, a signal to potential predators that the fungus might be poisonous or distasteful, or the bioluminescence might simply be a byproduct of the fungus's metabolism, a side effect of the chemical processes involved in breaking down wood. Not every trait needs to have an adaptive function. Sometimes biology just happens to produce something beautiful or strange as an accidental consequence of other processes. Interestingly, bioluminescence in fungi appears to be under circadian control in at least some species. Researchers studying a tropical bioluminescent mushroom in Brazil discovered that it increases its production of luciferin and luciferase at night, when the glow is most visible, and decreases production during the day when light would be wasted. This timing requires an internal clock, a way for the fungus to know when it's day and when it's night. It suggests that bioluminescence serves an important function for these fungi. something significant enough that they've evolved to regulate it carefully to produce light only when it will be useful. All known bioluminescent mushrooms use the same basic enzymatic pathway, suggesting that this ability evolved once early in the evolution of mushroom forming fungi and then was either retained or lost in different lineages over millions of years. The bioluminescent species that have been studied are all white rot fungi, meaning they can break down lignin, that tough woody substance in trees. Most grow on wood rather than soil. Some scientists hypothesize that the bioluminescent reaction might actually help protect the fungus from oxidative stress during the process of decomposing wood, with the light being a side effect of this protective mechanism. The oxygen molecules involved in breaking down lignin can damage cellular components. The luciferin luciferase reaction might help neutralize these dangerous oxygen species while coincidentally producing light. Mushrooms grow at rates that can seem almost magical. Most mushrooms grow much faster than plants because they don't grow through cell division like plants and animals do. Instead, they grow through cell enlargement. The cells of the developing mushroom inflate with water, swelling rapidly to create the mature fruiting body. It's like filling balloons with water, expanding existing structures rather than building new ones cell by cell. This is why mushrooms are over 90% water and why they need such humid conditions to develop properly. Without adequate moisture, the cells cannot expand and the mushroom cannot grow. Some mushrooms can grow from tiny pins, barely visible bumps on the mycelium, to full-sized fruiting bodies in just 24 hours. This rapid transformation is what makes people say mushrooms appear overnight. In a very real sense, they do. You might walk through a forest one evening and see nothing, then return the next morning to find the ground covered with mushrooms that weren't there the day before. This seemingly sudden appearance isn't magic, of course. The mycelium was there all along, hidden in the soil or wood, waiting for the right combination of temperature, moisture, and other environmental cues to trigger fruiting. Oyster mushrooms, Pleurotus species, are among the fastest growing mushrooms and are popular with home cultivators for this very reason. Under ideal conditions, they can go from spore to harvest ready mushroom in as little as 3 to 4 weeks. Other common mushrooms, like white buttons, typically take 6 to 8 weeks from inoculation to harvest. Shiitake mushrooms, prized for their rich flavor, take a bit longer, often 8 to 12 weeks, though this varies depending on the substrate and growing conditions. The bamboo mushroom, or veiled lady, Phallus induciatus, holds the record for visible growth speed. This tropical mushroom, found in warm climates throughout Asia, Africa, and the Americas, grows so fast that you can actually watch it happen in real time. It emerges from an egg-like structure in the soil and shoots upward, pushing out of the ground at a rate of about half a centimeter per minute. That's 3 centimeters every 6 minutes, or 30 centimeters per hour. At this rate, you can sit and watch it grow, see the stem elongating, the cap expanding, the delicate lace-like veil unfurling from beneath the cap. The tissues crackle and creak audibly as they expand, creating a sound like cellophane crinkling. Within a few hours, the mushroom reaches its full height of 15 to 25 centimeters. displaying an intricate white lattice skirt that hangs from the cap. This veil helps attract flies and other insects to the mushroom's foul smelling spore mass, aiding in spore dispersal. Mushrooms typically appear after rain because they need moisture to develop. Rain soaks into the soil, raising humidity levels both in the ground and in the air just above it. This moisture triggers the mycelium to begin forming fruiting bodies. The mycelium may have been present in the soil for months or years, growing slowly, feeding continuously, waiting patiently. When rain soaks the ground and humidity rises and temperatures are in the right range, the mycelium responds by initiating fruiting body formation. Chemicals signal to the mycelium that conditions are favorable. Resources stored in the mycelium mobilize, flowing toward the sites where fruiting will occur. Within hours or days, mushrooms emerge, seemingly out of nowhere, pushing through leaf litter and soil. This sudden appearance after rain has contributed to mushrooms' mysterious reputation throughout human history. Before people understood the biology, before they knew about mycelium and spores, mushrooms seemed to appear by spontaneous generation, as if the rain itself created them. This seemingly magical quality, combined with their sometimes hallucinogenic or poisonous properties, made mushrooms objects of wonder, fear, and reverence in many cultures. Different mushroom species have different lifespans. Some mushrooms last only a day or two before shriveling and decomposing. Others can persist for weeks or even months. The fruiting bodies of some bracket fungi, the shelf like mushrooms that grow on trees, can last for years, adding new layers of growth each season, much like tree rings. However, the mycelium of the fungus lives much longer than the fruiting bodies, potentially surviving for centuries. Fungi can survive in extreme environments. Some species live in the soil of Antarctica. Others thrive in the intense radiation zone around the damaged Chernobyl nuclear reactor, where they actually use melanin pigments to capture energy from radiation, a process called radiosynthesis. Desert fungi survive by remaining dormant during dry periods and activating only when rare rains arrive. Deep sea fungi live in the ocean's abyssal zones. Fungi have even been found growing inside active volcanoes and in radioactive waste sites. This remarkable adaptability has made fungi valuable to humans in unexpected ways. Fungi can break down many pollutants and toxic substances, a capability called bioremediation. Some fungi can degrade petroleum products, helping clean up oil spills. Others can break down plastics, pesticides, and even some heavy metals. Researchers are exploring ways to use fungi to clean up contaminants